Hello, good morning everybody. Hopefully you can all hear me, and welcome to today's webinar from the Francis Crick Institute. And today we're going to be talking about using flow cytometry to look at multicolour experiments, so looking at high dimensional panel design. So those of you who have not been to one of these webinars before, let me introduce myself. My name is Derek Davis. I am the STP, or Science Technology Platform Training Lead here at the Francis Crick Institute in London. But for many years I ran the core flow cytometry facility there, and you can follow the Crick flow cytometry facility on Twitter, or you can also follow my training account, so that will give you information about upcoming webinars. One thing I should say is this webinar will be recorded, or is being recorded, and will be made available to all attendees afterwards. But please hang around, because you do get the chance to ask questions. So hopefully all you saw on the splash screen when you came in, there's a Q&A box. So if you have any questions as we go along, please write them in there. I'll probably deal with those at the end if that's okay. If there's any issues with sound, or if you want me to slow down or anything like that, please put that in the chat box. I'll try and keep an eye on that as we're going along. So what I want to cover today in an hour is the basics of panel design, multicolour panel design, because I imagine many of you here are doing simple experiments, 1, 2, 3, 4 colours or something like that. But the great advantage that we know of flow cytometry is that we can multiplex. We can put as many different fluorochromes as we have detectors on our cytometer. So it makes it a very useful technique if we're looking at a very heterogeneous population. The more markers I can add to a cell, the more specifically I can define that subpopulation. But all of that comes at a little cost. We're going to need to think about the two things that we always need to think about: our tools, our fluorochromes, and our cytometer. We're always going to be limited by what we have at our availability. I'm going to talk a little bit about sample preparation, although that tends to be specific for particular cell types. And we're going to look at some rules. I always put rules in inverted commas because there's almost nothing that can't be broken. But what I want to be able to do this morning is give you the overarching principles, which would be applicable to a 2, a 3, a 4 colour experiment as much as they are to a 22, 23, 24 colour experiment. Because we're trying to optimize being able to see what we want to see, or what we think we're going to be able to see. And as I say, I'm not going to be very specific with the design here today. We're not going to design a 25 colour panel today, but by giving you some tips and tricks, hopefully you can go away and start thinking about how you could do that. So we know that flow cytometry has a very simple definition. Basically, it's making measurements on cells in suspension. And importantly, remember, we can do this at several thousand cells per second. Makes it much more robust than microscopy, for example. So we can get very statistically robust data sets, very useful if we're looking for rare events. And we're making measurements on our cells or particles one by one. Now, flow cytometry is a single cell technology. And those measurements that we're making are physical and chemical. So by physical, cell size, cell shape, cell complexity. By chemical measurements, we mean by adding fluorescence. That fluorescence is going to light up what we're looking for, whether that's an antigen, a protein of some sort, whether it's a functional aspect of the cell, whether it's something like DNA or RNA. Now, those of you who have been to these webinars before might be familiar with this slide, and I make no apologies for putting it in again, because when we're teaching, it's one of the most important slides to think about, because whatever experiment we're doing on a flow cytometer, say whether it's something simple or whether it's very complex, we need to bear these same points in mind. And the first of those is what we call signal to noise ratio. A signal is what we're actually trying to measure. So it's a combination of our fluorochrome, the brightness of our fluorochrome, and the amount of target that we have on our cell. Now, flow cytometry is a measuring system, so it's always going to have some noise, some background. And we're always trying to raise our signal and decrease our noise. So if I'm doing a 1 colour experiment, that noise comes a little bit from background noise of the cytometer, but mostly from autofluorescence of my unstained cells. So that's really all I have to consider. Whereas if I'm doing a multicolour experiment, that noise also comes from spillover of other fluorochromes in the system. So my signal to noise ratio is always going to be worse the more fluorochromes I add. And that's going to affect these two things. Now, sometimes these two terms are used almost interchangeably, but I see them as slightly different. So by sensitivity, I mean, can I detect, or can my flow cytometer detect something that's weakly positive above background? By resolution, I mean how well can detect it above background. And that's something that becomes very important when we start thinking about multicolor panel design. And then we also have data spread or measurement error. Flow cytometer is a measuring system. It's never going to be perfect. There'll always be some error in the measurements we make. But that error comes from multiple sources. It comes from biology. We know that there's a great variation in the expression of markers, for example. It can come from what we do to our samples. Sample preparation, fixation, permeabilization, buffers. They can all affect our cells and our fluorochromes. It can come from the cytometer. Hopefully we're all aware that if we press high on our flow cytometer, cells go through more quickly, but we get lower resolution data because we're making more error in our measurements. And it can also come from data analysis. So try and always think about these things when we're designing experiments. So I'm probably teaching everybody here to suck eggs, but this is how a flow cytometer works. We take our cells in suspension, we label them with some fluorochromes, we pass them through a light source, get fluorescence emitted, which we pass through some optical filters, capture that light with an electronic system, and quantitate it. So the great thing about flow cytometry is we can get numbers, we can get quantitation. And we know that we can use a single laser to excite multiple fluorochromes, or if our cytometer has multiple lasers, we can excite multiple fluorochromes, and from each of those we can capture and quantitate the signal. So this makes flow cytometry fantastically useful if I have a heterogeneous population. And it's like a big biological game of guess who, isn't it? The more things I can measure on my cells, the more specifically I can define them. And that's something that's changed a lot in cytometry over the years, because 40 odd years ago, we could do this, one color analysis or two color analysis. So here we could separate our CD8 and our CD4 cells. And there's no reason why a one color experiment today isn't just as valid. We do a lot of DNA analysis, single parameter histograms, GFP fluorescence, screening. Those experiments are no less valid than a high dimensional multicolor panel. But we know that in recent years, we've seen a whole new set of lasers become available, a whole slew of fluorochromes and fluorescent proteins, which means that we can do this. So this is an example from one of the first 17 color flow cytometry panels that came from the Vaccine Research Center at the NIH. And this was published, I think, in 2005. So that's quite a long time ago. But to be honest, in our lab, we'd have very few people who are doing more colors than that now, even though it's possible. And the reason is because it's not easy. But you show people that sort of data and their first response is, I want to be able to do that. And my first response to them is, I usually, or unless you really want to, you shouldn't overcomplicate things, because the more colors you add, the more fluorochromes you add, remember, the lower the resolution becomes, because our background, our noise is starting to increase. So you need to really think about your experiment. So if you're doing a one color GFP experiment, it's very easy, isn't it, to see the GFP positive cell against the relatively bland background. But what happens when I add more fluorochromes into that mix? Those GFP cells are still there, but it makes it maybe a little harder to see them. So we've got to be sure that a multicolor panel is really what we want to do. Okay, let's assume we've got to that point. Where do we actually start? So, as I said at the beginning, we're always going to need to think about these two things, our instrument and our fluorochromes. So we can help you with that in a core facility. However, this is where you're going to come in, because you know about the biology of your system. You understand your antigens or your markers. And then these two things together, or the tools and the biology, are going to help us maximize the resolution, remember, to be able to see what we want to be able to see. So we're always going to be limited by this, aren't we? The configuration of the flow cytometer. Now, if you're lucky enough to work in a core facility, you may have a whole range of cytometers, from simple ones right up to high-end ones. And there are two cytometers on the market at the moment, the Becton Dickinson Symphony and the Biorad ZE5, that are capable of measuring 28 different fluorochromes. But let's take a look at this cytometer here. Now, attached to all of our machines at the Crick and downloadable from our website are the configurations of the cytometers. So this tells me what I need to know before I even start thinking about designing an experiment. It tells me how many lasers I have available. So here I know that I've got 5 lasers, and it tells me the colors. So I've got a blue, yellow, green, a UV, a red, and a violet laser. But it's not just about the lasers, it's about the number of detectors we have on each of those laser lines. So I can see here I have 2 detectors on my blue laser, 4 on the yellow-green, 2 on the UV, and 3 on both the red and the violet line, which I think adds up to 12 colors. Is that right? Something like that. So I know I'm going to be limited immediately to the number of detectors that I have. So I know that if I want to do an experiment that involves the brilliant UV dyes, I can only measure 2 of them. This also tells me which optical filters I have available in my flow cytometer. And again, this is going to be important to me. Most of the time we don't have to change these, but again, taking that example, if I'm using 2 BUV dyes, it may be that these aren't the appropriate filters for that. Now I can change them, but I'm still limited to 2 dyes. And then this column here on the right tells me which fluorochromes I can measure in each of those detectors. Now I may know that, particularly if you're experienced, you would know that FITC is always going to be measured in this detector, but it gives you some examples. Often people forget where, for example, APC Cy 5.5 might be measured. So as well as knowing something about the cytometer, we of course need to know something about the fluorescence and the fluorochromes that we're going to use, because flow cytometry is a fluorescence-based technology. So we can go to the internet and we can look at those spectral viewers, which tell us the excitation and the emission spectrum. So can I excite this fluorochrome? And if I can, which filter would I use? They don't tell us anything about brightness, so we need to know how bright our fluorochromes are. When we start thinking about multiplexing, we're going to need to know how similar our fluorochromes are to each other, because obviously the more similar they are, particularly in terms of their emission, the harder it is to be able to use them together. And we may need to know how stable those dyes are. That may be affected by our sample preparation. So if you came to our introductory seminar, we looked at some of the classes of fluorochromes that we can use. So things like small cyclic ring dyes like fluorescein, big proteins that are fluorescent, things like PE, APC, the whole range of fluorescent proteins like GFP. So these small dyes we know are probably going to be relatively resistant to fixation, so that helps us with our experimental design. These larger protein dyes are maybe less resistant to fixation, but they're possibly brighter. We know that we can also use tandem dyes, which are a combination of a large protein donor dye and then a small fluorescent acceptor dye. Again, because this is a chemical coupling, it can be affected very much by fixation. And then we have things like the Q dots and more recently the polymer dyes. So we don't have to use one class of dye the whole time. We can of course mix and match between these, but knowing something about their structure and their behavior is very important, as is of course knowing this, the spectral viewer that I mentioned just now. So many of you will be familiar, or possibly even fed up with seeing this histogram, this plot here. This is the excitation spectrum and the emission spectrum of fluorescein, FITC. And we know that we can optimally excite it at 488 nanometers, and this is its emission wavelength. But remember, we don't collect all the photons on a traditional flow cytometer, we just collect a portion of them, and that is defined by the optical filter that we have in there. So generally something like a 530-30 filter to allow us to capture the maximum emission of fluorescein. Now, if I'm just doing a one-color experiment, of course, I'm exciting fluorescein at 488 and I'm capturing its fluorescence. However, this spectrum also tells me that fluorescein will be excited by a violet 405 laser nanometer. Nowhere near as efficiently. In fact, it may take 20 times as many photons of 405 nanometer light to hit that fluorochrome before I get an excitation of an electron, but nonetheless, it does excite it. And if I've got a high-powered violet laser and I've got a fluorescein labeled antibody that's directed against a high density antigen, something like CD45, although the efficiency of excitation is low, the actual number of photons may be relatively high. Now, the problem with that is that if I've got the same detection filter on my violet laser, some of those photons from FITC will be collected in it. Because no matter which laser wavelength we use to excite fluorescein, the emission spectrum is always going to be the same. Now, of course, I know this and I can do something about it because I will have a single color control and I can account for or compensate that overlap in exactly the same way as I do any other compensation. But as we'll see, it causes problems because I'm ending up with photons that I don't want in a detection channel that I'm using for some other marker. The spectral viewers are also very useful for our tandem dyes. So remember, the problem that we have with tandem dyes is that we're always going to see fluorescence from the acceptor dye. So this is P size 7, so this is size 7 emission. But we're always also going to see some emission from the donor dye, no matter which manufacturer we get our PE size 7 from. There will be slight differences. There's manufacturer variation, there's a lot variation. And we know that there's also variation over time. Over time, we see more of this donor fluorescence becoming seen, visible. And there's also a change with fixation. So we always have to be a little bit more circumspect with our tandem dyes. And the only thing I'll say about tandem dyes is, PE Sci 5 here. I know that PE Sci 5 is a tandem dye. PE is a fluorochrome. Sci 5 is a fluorochrome. A little hyphen in the middle tells me it's a tandem dye. BV786 is also a tandem dye, but I wouldn't know that from its name. But if I go to the spectral viewer, I can see that. So here's my fluorescence from, in this case, I think it's dye light 786, which is the acceptor dye. But we see that residual fluorescence from the donor dye, which in this dye is brilliant violet 421. So as long as I know that these dyes are tandem dyes, I would treat them with the same respect that I would treat any other tandem dye. And by the time we start getting above 6 or 7 colors, we are going to be having more tandem dyes in our experiment. Of course, the more I add, the more we have to be wary because of this residual fluorescence. So tandem dyes are always going to emit light in 2 different areas of the spectrum. And that again is going to impact on our resolution. But these spectral viewers, remember, don't tell us anything about the brightness of a fluorochrome. They do tell us the excitation and the emission characteristics, but I wouldn't know which of FITC and PE size 7 was the brighter dye. In order to judge that, I need to know these 2 things: the extinction coefficient and the quantum yield. Now, the extinction coefficient is a measure of how good a molecule is at absorbing light. If I throw 100 photons of light at a molecule, I don't always get 100 excitations of an electron. And the quantum yield is, if I do get 100 excitations of my electron, how many times does it actually emit a photon? It doesn't always happen. Now, we can find those figures on the internet, but they can be slightly misleading because the way they're normally derived is to take the solubilized fluorochrome, excite it at its maximum wavelength, which generally we don't do in a flow cytometer. We excite it somewhere near the maximum, but not the exact maximum. And then you collect all of the light that comes out of the fluorochrome. Again, something that we don't do on a traditional flow cytometer. We just collect a portion of the light. And remember, what we're doing in our flow cytometry experiment is we're taking a fluorochrome, we're binding it to an antibody, let's say, which is binding to its antigen, which is present on a cell which has a level of autofluorescence. And that autofluorescence will be different in different areas of the electromagnetic spectrum. So the way we assess brightness in a flow cytometer is to use this thing called a stain index. Okay? And we do that by looking at the fluorescence from our dyes under the conditions that we're actually going to use them in our cytometer. So exciting them with the laser that we have on the cytometer, collecting them through the optical filter that we have on our flow cytometer, and also taking into account the background autofluorescence. So this is going to combine those two things, resolution and sensitivity. So how do we derive this stain index? It's actually fairly simple. I take a fluorochrome labeled antibody, and in this cartoon I can immediately see that I've got a positive population and a negative population. Flow cytometry is great because we get that visualization, but I also want to get a number. So I could just look at the distance here, or the difference between the mean or the median fluorescence intensity of the positives and the median fluorescence intensity of the negatives. But remember, autofluorescence is different in different areas of the spectrum. So what I really want to know is this sort of distance, the distance between the end of the autofluorescence and the beginning of the positive fluorescence. So I'm going to penalize this distance, if you like, by dividing by the spread or the width of this negative population. In fact, I divide that by 2 times the robust standard deviation. Now, as I'm sure you're all aware, standard deviation is a measure of data spread, and it's normally statistically derived by looking at the distance of each point from the mean of the population. The robust standard deviation is using, rather than the mean of the population, the median of the population. So it's derived by looking at the distance of each point from the median rather than the mean. Again, we do that because the median is a little bit less influenced by outliers than the median. So I can calculate that stain index, and that will give me a number. Now, the reason why that penalization, if you like, is important, because let's look at a very similar situation. Here, again, I've got a positive and a negative. The distance between the mean or median of the positive and the negative is the same, but in this area of the electromagnetic spectrum, the data spread in the autofluorescent cells is much bigger. So I've got less distance here to see something that's weakly positive. I've got a lower resolution. So that stain index will be lower, will be higher in this example than it exists this one. So that tells me that this fluorochrome is a relatively brighter fluorochrome than this one. And we can do that for every channel that we have in our flow cytometer. So this is a 8-color cytometer, 2 violet detectors, 2 red detectors, 4 blue detectors. And again, immediately I can see, so what we've done here is looked at CD4 in every fluorochrome that we can measure. So I can see in each case I've got a positive and a negative population. But I can also see that in my APC, my PE, particularly maybe my BV421, my positives and my negatives are much further apart. I can also see, though, that in some areas of the spectrum, I get very low data spread in my negatives. Some areas I get very high data spread. But then for each of these parameters, I can calculate the stain index and get a number, so I can start to rank my fluorochromes. I put this here. This is actually a very important point. Maybe I shall put it in red. This stain index is dependent on conditions. So in an ideal world, what I should do is do this stain index on surface stained unfixed cells, then do it again after I've fixed my cells in ethanol or formaldehyde. Then I should do it again on an intracellular marker. Because remember, our conditions can potentially affect our fluorochromes, and they could potentially affect the stain index. So sometimes, particularly if I'm doing a combination of surface and intracellular markers, I may want to do this specifically for my panel. But luckily we can come up with this sort of ranking table. So this is from BioLegend and this one's from BD. In both cases, they've sort of rather qualitatively assessed the fluorochromes. So they've ranked them from very bright to dim in this case, or given them a score out of 4 in this case. I prefer to do it that way, actually, rather than using the absolute numbers, because that absolute number does depend on the actual cytometer, the laser power, the filters, and indeed the electronic system, the measuring system. So ranking them qualitatively is probably a good way to do it. But this now means that I can start thinking about my experiment, because if I'm looking for a marker that's weakly expressed, or I don't know the expression level, I'm going to choose a very bright fluorochrome. If I'm doing a 2 or 3 or 4 color experiment, I've got half a dozen or so very bright fluorochromes, so I could use a combination of those. But once I start getting to the realms of 15, 20, 25 colors, I don't have that many very bright fluorochromes, so I'm going to have to use some of these dimmer ones. So this is where we're going to have to start thinking about not just the brightness of the fluorochrome, but the antigen density, or the density of the target that we're looking at. And so why do you need to even think about the fluorochromes? Okay? Because it's going to be very important. Some of your probes you can't change the fluorescence of, for example, so you're always going to be working in one area of the electromagnetic spectrum. So, for example, when we're looking at apoptosis, one of the dyes we use is a dye called TMRE. We can't change the fluorescence of that as we can with antibody staining. So we're always going to be limited to an area of the electromagnetic spectrum. We know that some of our fluorochromes, particularly the tandem dyes, may require special handling. And we know that we need to know something about the emission, the excitation and the emission of our fluorochromes. And we're always going to have to be thinking about sample preparation. Remember, our fixation, our permeabilization, our buffers may affect our antigens and our fluorochromes. Of course, it may depend on the location of our antigen. If we're doing surface staining the whole time, our life is easier than if we're looking for something that's in the nucleus as well, for example. And we're always going to have to be aware of cellular autofluorescence. So I spent a bit of time before we even start really going into the nitty-gritty of panel design. So, how do we do it? What's the approach? What's our journey? And it's always going to start actually by sitting down and thinking, rather than just going to the fridge and seeing which antibodies you have. You need to define the biological question that you're going to ask. Excuse me, I've lost my cursor there for a second. There it goes. There it's back. You need to always think about the biological question. Where is my antigen? So if it's on the surface of the cell, it's a lot easier than if it's in the nucleus. Do I need any special buffers? Some of our fluorochromes, remember, are affected by pH. We may need to think about that. We're going to need to understand the things that we're looking for, our antigens or our markers. Are they on lots of cells, or are they only on a few cells? Are they very highly expressed, or are they weakly expressed? Or is there a gradation in the expression? Is there a variation in the expression? Knowing these two things helps us to start thinking about maximizing our resolution. And resolution is probably the most important thing we're going to think about in multicolor design. So we've already started thinking about assigning our brighter fluorochromes to our weaker antigens or our rarer antigens to give ourselves the best chance of being able to see it. And let me plant this seed here now. We're always going to be aware of antigens that are expressed on the same cell. Remember, flow cytometry is a single cell technology. If I'm doing a 16-color experiment and actually only one of those markers is expressed on each cell, my life is a lot easier than if I have that heterogeneity where some cells express 1, some express 2, some express 3, some may express all 16. So knowing something about that expression will also help you. And it's not going to be easy. There's no getting around that. If you want to move from a 2-color to a 20-color panel, it's going to take some time. And we may sit down and think of an experiment that looks on paper like it will work, but of course we need to test that. We're never really going to know till we get that to the cytometer. And it's almost certainly never going to work first time, so we're going to have to be prepared to take the time to iterate that, to keep changing it until we get to the point where we can see what we want to be able to see. So we could choose to do an experiment like this, couldn't we? If I've got a cytometer with a blue laser, I could choose to look at 4 colors, FITC, PE, PSI5, PSI7. They're all excited by the same laser, and they all have different emissions. But if I'm looking at the PE channel here, all of those other 3 fluorochromes in the system will produce photons that end up in my PE channel. So it's not just the PE itself. Some of those photons, because of the emission spectrum of fluorescein, will be detected in the PE detector. And some photons from PSI5 and PSI7 will also be detected in the PE channel. Remember, residual fluorescence from our donor dye. So even with this simple 4-color experiment, I'm losing resolution, particularly in my PE channel. So I could make my life a lot easier by using that cytometer I showed you earlier. So let's use 4 different lasers. Let's use a violet laser, a blue laser, a yellow laser, and a red laser. Spread my fluorochromes out. I won't necessarily have no problems, but I'm probably going to have fewer problems. And this is one of the great things about multicolor or multi-laser cytometers. I don't have to use all the detectors. I can design a more flexible experiment. But this is just thinking about the fluorochrome, isn't it? Remember, we also have to think about autofluorescence, and that varies in different areas of the spectrum. I've added this all together here. This isn't going to be in every cell. Every cell will have its own unique autofluorescence signature. So if you're dealing with plant cells, we know that chlorophyll is particularly autofluorescent in the red end of the spectrum. So by knowing that, if I'm trying to label something with another fluorochrome, I'm more likely to use a fluorochrome that emits in the blue end of the spectrum. Melanocytes are very fluorescent in the sort of purple, indigo, I suppose you'd call it, range of the spectrum. If I know that, I might use a red fluorochrome to detect what I'm looking for. You'll notice there's actually quite a clean area here, around about 600 nanometers. But I said this is very general. You might have to check that for your own cells. But knowing that, again, helps you design your experiment, helps you keep away from the areas of high autofluorescence, because autofluorescence is going to induce noise. So the question, of course, is, how big does your panel actually need to be? And as I said, it's always tempting, isn't it, if I've got a 20-color flow cytometer, to try and design a 20-color panel. But as we hope I've shown you already, you're getting the impression that the more colors we add, the problem is we start losing resolution, and that means our dim signals are harder to see. So if we go back to that example of the PE channel I showed you with that 4-color experiment, if we think of the PE channel as a sort of rain catcher, okay, if I run unstained cells, I'm going to have a level of autofluorescence. Remember that's good because it gives me a background to see something that's positive. So if I'm doing a single color PE experiment, I've got this much space in my PE channel to detect something that's positive. But if my cells also have fluorescein on them, remember some of those fluorescein photons will end up in that PE channel. My cells also have psi5. Some of those psi5 photons end up in the PE channel. If I have psi7, some of my psi7 photons end up in the PE channel. So although we can account for that overlap by the process of compensation, we don't physically remove those photons. So what it means is I've now only got this much space to see something that's positive. So I've lost resolution simply by adding these other fluorochromes. So I said there are some rules, and I said I always put rules in inverted commas because there's nothing that you can't break. But when we start thinking about multicolor analysis, these are the things we should think about. So first of all, let's try and spread our fluorochromes across our laser. So that one laser system we have, if I've got a blue laser, but I've got 5 detection channels, and I want to do a 2-color experiment, let's use the first detection channel and the last detection channel. The further they are apart, the less overlap I'm going to get between the fluorochromes, the better my resolution is going to be. Of course, a 2-color experiment is not that complex. So if I've got more colors, let's start thinking about spreading my fluorochromes across lasers, particularly, again, if I've got co-expressed antigens, because I'm going to get less overlap if my fluorochromes are on different lasers. Not necessarily no overlap, but less overlap. As we've seen, try and put our weakly expressed markers on our brightest fluorochromes, or in areas of the spectrum where we get lower background, lower autofluorescence. And then try and avoid channels where we have large spillover. Now that sometimes is going to be impossible. We may have to use all the detection channels, and we know when we have 2 detection channels that are quite close together in the electromagnetic spectrum, we're going to have a lot of spillover. But we can use that to our advantage, as we'll see, I hope, a little bit later. So, that 2-color experiment, CD3, CD8, we know that they're co-expressed, and I've only got one laser. So to maximize my resolution, I'm going to use fluorescein and psi7, either end of that laser. If I'm lucky enough to have a 2-laser system, I can make this experiment even better. I can have one of my analytes on the first detection channel of the blue laser and the other on the far end of the red laser. Not only are they now spread out as far as possible in the spectrum, but they're on different spatially separated lasers. Actually, this sort of cartoon layout is something that's very useful. So in the lab, we have these A3 laminates of the layout of each of our flow cytometers. So this is a sort of example of that 5-color cytometer I showed you earlier on. We've got a UV, violet, blue, yellow, green, and red laser. And this represents the amount of space we have to detect emitted fluorescence. So obviously with the UV, I've got pretty much the whole of the electromagnetic spectrum. So if we want to do a 5-color experiment with that cytometer, again following one of those rules, we've got 5 lasers and 5 parameters. Let's put 1 parameter on each laser beam. So by using the first channel, and the first channel is less likely to have overlap from things that are further up the spectrum, but here we don't have them. So I've spread out my lasers across, I spread out my fluorochromes across those lasers. Very easy. So you can imagine that I could quite easily go to 10 or 12 by using the other end of each of those laser beams and see if there's any space in the middle. So here, 10 colors. So we've got those same 5 colors at the beginning of the laser line, of the space from each laser, and then the other 5 colors at the end. And you can see, particularly with the UV and the violet, there's quite a lot of space between those. So I could quite easily add in some fluorochromes here. Of course, this is just looking at the fluorochromes. We're not taking into account any antigens at the moment. The only slight worry I would have with this is, remember I said earlier on that all fluorochromes can be potentially excited by all laser beams. So one thing I always look at is this. So these 4 fluorochromes in particular are all emitting in the same area of the electromagnetic spectrum. So although this is optimally excited by UV, this is optimally excited by violet, and so on, there may be some excitation of this dye by the violet laser, and that would be detected in the same area of the spectrum. So that may potentially give me some problems. So I'm always looking at that as well. So similarly, sort of here, we may have that problem. But it's not just about space in the spectrum, is it? It's about, remember, it's not just about the fluorochrome, it's also about the biology, the antigen density. So as well as thinking about that fluorochrome brightness, we also need to think about how many markers we have and what are the staining patterns. So what do I mean by those two things? Well, the antigen density is how many targets we have. So how do you find that out? So if you're lucky enough to be dealing with peripheral blood, if you're looking at CD markers, you can get that information from multiple websites. Again, this is from Becton Dickinson, and it tells me what I need to know. It tells me how many targets I have. So I know that CD3, CD4, CD8 are very highly expressed, whereas I know that CD25, CD127, for example, are much less highly expressed. There may only be a few hundred or a few thousand targets in those cells. So I'm going to have to fight harder to see those. So I'm more likely to put a bright fluorochrome on these lower expressing markers than I am these highly expressing markers. I don't necessarily want to use a very bright dye on something that's highly abundant. But of course, not everybody is dealing with human CD markers. So, how do you find out what your antigen density is? Well, if you're lucky, it's been published. The manufacturers may be able to tell you that with their data sheet, or it may be available somewhere on the internet, or you may have that knowledge in your lab or personally. If you don't know, you have to do it yourself. Okay, and of course we can do this by flow cytometry, and the way we do this by flow cytometry is using calibration particles. So there are a couple of kits out there that allow us to do that. This one here is a kit called QuantiBright from Becton Dickinson. There's also a thing called the Quiffy kit from Dako. And what these are a set of multi-peak beads, and with the data sheet you get from those beads, you'll get a value for the equivalent amount of fluorescence for each of those peaks. So QuantiBright beads are PE labeled. So we have 4 different levels of intensity of PE. So on our optimized flow cytometer, we can run the beads, we can calculate the median fluorescence intensity for each of them, and then we can plot that against the receptor number, which is on our data sheet. And we get a nice regression curve, which is useful in 2 ways. One, it tells me that my cytometer is very linear because I'm getting an R² value of close to 1. But now I can take my PE labeled cells, run them at the same voltage, calculate the median fluorescence intensity, and read off approximately the number of receptors that I have there. But remember, we always have to think about biology, don't we? Remember, I said biology is one of the biggest variables. So if we look at this plot here, so these are, in this case, 5 populations of beads, and the red hatching here is my cells. So you can see, although the median fluorescence intensity tells me that there's about 60,000 markers on this cell, it varies anywhere up to 500,000 down to about 10,000. There's a huge variability. So it's not just about the number or the average number of markers. It's also about looking at the expression pattern that we get. So how can we think about expression pattern? Well, we can classify our expression pattern in 3 ways. What we call primary, secondary, and tertiary. And what do I mean by those? Well, primary is something like this. My marker is on or it's off. It's there or it's not. Something like CD3. It's on a T cell, or it's defining a T cell, or it's not present on things that aren't T cells. So if I'm getting that very binary expression pattern, I probably don't need to think too much about which fluorochrome I'm going to use, because I just want to be able to separate my positives from my negatives. It may not matter that they're as far apart as I can possibly get them. But remember, you're always in control of your experiment. This is what we call a secondary pattern. So I am getting some very bright cells, as we saw in the previous slide, but I'm also getting some cells that are maybe only just positive, just above the background. So in that situation, I'm probably going to want to use a very bright fluorochrome. I want to bring these weakly positive cells as far to the right as I can. So I want to use a bright fluorochrome. I'm sure you've all seen expression patterns like this as well, where we get a shoulder or maybe get a small number of weakly positive cells. Again, in that situation, I want to use a very bright fluorochrome to give myself the maximum chance of being able to see that. If we're dealing with antibody staining, another thing we might want to think about is titration. Well, I'm sure we want to think about titration. And we've all been taught, haven't we, to titrate our antibodies, and there are multiple ways of doing that, or certainly multiple ways of plotting it. The idea being that we should be running our sample at the optimal titer to give us the best separation. So optimal titer here would be somewhere here, I suppose, to give us the best separation of our positives and our negatives. And if I'm trying to quantitate the number of receptors, that's exactly what I should do. But again, you're in control of your experiment. That would be a saturating titer. There's also this concept of a separating titer. So even if I come down to here, for example, I could still separate my positives and my negatives. Maybe not as well as at the optimal titer, but I can still do it. And maybe that's all I need to be able to do. And actually this can also impact on your experiment as well, because the brightness of our particular fluorochrome is going to affect how much of that fluorochrome spills over into other channels. So again, you may be familiar with these 2 terms, spillover and spread, which again are slightly different. So we measure spillover by compensation, and we know that some photons from almost every fluorochrome will end up in almost every other detection channel. And we account for that spillover by the process of compensation, by using a single color control. But we also know that when we set compensation, I'll show you here, we reveal this data spread. And that spread is measured by the standard deviation, as we saw earlier. It gives us a measure of how far apart everything is from the median. So we have a great data spread here. And that spread is dependent on the amount of spillover, but also the antigen density and the relative fluorochrome brightness. So the brighter this population here becomes, the more spread out it becomes. So that helps us again when we're thinking about our experimental design. So let's look at these 2 plots here. So this is a combination of PE and BV421. So we can see we're getting relatively low expression of our BV421, but no matter how bright our PE becomes, I can still separate that. I'm getting very good resolution. Now if I simply change my fluorochrome, keep PE the same, but now change it to PE Cy5, I know I'm going to get more overlap because by exciting them by the same laser, I know that the PE part of PE Cy5 will end up in my PE detection channel. So now by the time my PE population is very bright, I'm losing resolution. I can no longer optimally distinguish my weakly positive PE psi5 population. So this is the concept of having that separating titer. If I don't have to have my PE as bright as this, I could potentially run it here, which means that I get my resolution back again. So this is going to become important in any multicolor experiment, but I want to be able to get a number for that, don't I? I want to get some metric. I want to assess how much resolution I could potentially lose with a combination of fluorochromes. And unfortunately, it's very difficult for me to be able to tell you that in a seminar like this, because it depends on multiple things. It depends on your cytometer. Not all our cytometers have the same lasers or the same laser power. They don't all have the same optical filters. It's also going to depend on which other fluorochromes you have in your panel. Remember, every fluorochrome could potentially leak into any channel. So it depends how many you have and which you have in there. And it also depends on the expression level of those fluorochromes, because as we know, as our expression level gets brighter, we get more data spread. So there's another cartoon that says this in a very similar way, I suppose. If we're looking at a very simple two-color experiment, I've got a double negative population, a double positive, and then two single positive populations. Now, if I'm just looking at these two, something that's negative for fluorochrome 1, I've got this much space, haven't I, to see something that's weakly positive. Whereas if I'm looking at things that have been stained with both of those fluorochromes, because of this data spread I get here, I've now only got this much space to see something that's weakly positive. So I get some loss of resolution. I want to be able to assess that loss of resolution, and there are a couple of ways of doing that. One's very complicated, which I'm going to show you first, and then there's a couple of simpler ways of doing it. So that paper I showed you earlier, or that what's the word? Figure I showed you from that paper earlier. There's another paper from Guyan a few years ago now that shows you how they derived the combination of fluorochromes that they used. So you want to calculate or assess the loss of resolution in each channel. So what they've done here is taken beads, in this case FITC beads, and correctly compensated FITC out of PE. And so we know we're correctly compensated because the median of these two populations is in the same place. But if, for example, we just look at these negative population, if I look at a histogram of that negative population in the PE channel, it would look like that. Whereas if I look at a histogram of the positive population, it would look something like that. So the medians are in the same place, correctly compensated, but we're getting much more spread in this positive population than we are in the negative. So we want to get a number for that. So what they did here in this situation was look at the 90th percentile of the negative population, the 90th percentile of the positive population, and looked at the spread, the number of channels that are different between those two. But remember that as the FITC gets brighter, we get more spread. We sort of penalize that by looking at the difference between the median of the positive population and the median of the negative population in the specific channel. But then we can come up with a number. You don't need to worry too much about how we calculate that, but it's, the spread of this divided by the brightness. And then you can come up with quite a complex picture that looks like this. So this is the 17 colors that we're using in that experiment. Let me walk you through this to make it a little bit easier. If we look at this column here, this is the amount of error that each column receives from other fluorochromes in the system. So what we're looking for is low numbers. The lower the number here, the less photons I'm getting, or the fewer photons I'm getting from other fluorochromes in the system. So that makes these channels good candidates for looking for dim markers, because the background, the noise, isn't going to go up very high. So Cascade Blue, very dim fluorochrome in itself, but almost no other fluorochrome in this system contributes error. Fluorescein, generally a reasonably dim dye, but again in this system, not many fluorochromes contribute error, which may make it a good candidate for a dim marker. Let's look at the column at the bottom here, which is the total error that's contributed by each dye. And if we look at Percy psi 5.5, it's hugely promiscuous, if you like. Its photons are ending up all over the place, not just in the channel we expect, but lots of other channels as well. Doesn't mean to say it's a bad dye. And of course, in this situation, they had to use that channel because they had a 17 color panel, a 17 color instrument. So again, it comes back to intelligent panel design. If I could use that channel, say a dump channel, or put my viability marker in that channel, the first thing I'm going to do in my analysis is remove all cells that are positive for that fluorochrome. And remember, flow cytometry is a single cell analysis. So if I remove the cells that are psi 5.5 positive on the cells I'm interested in, because it doesn't have that fluorochrome, we're going to see less loss of resolution. PE, very bright fluorochrome, remember? Not in this system particularly, or not a very good fluorochrome for a dim marker, although PE is bright. A lot of other fluorochromes are contributing error into the PE channel. So obviously that's a lot of work doing it that way. We can do it maybe slightly simpler by going back to the cartoon I showed you a few slides ago and deriving a stain index. So if I take the fluorochrome 1 negative population, I can derive a stain index. Remember, the difference between the positives and negatives divided by the standard deviation of the negatives. And then I can do the same when I've got both dyes in there. So I can then assess that loss of resolution by a very simple equation. So looking at the difference in the stain index of the double positives and the single positives, given as a percentage. So if there's no difference, if I see no spread, that will have a value of 0. So I'm getting no loss of resolution. So the higher that number, the more resolution I'm using. So then for each of your cytometers, you can produce what we call a resolution impact matrix. Okay, so this is now color coded, but we read it in very much the same way that we did that 17 color experiment. So if I look at FITC here, no matter which other fluorochrome is expressed alongside FITC, I don't get very much loss of resolution, again indicating that it may be a reasonable fluorochrome to use for a dim marker. Whereas psi 7, virtually every other fluorochrome in this system contributes photons, to the psi 7 channel, so I'm going to lose much more resolution. Again, looking at it this way, this tells me which fluorochromes are bad to use, in that they're going to contribute photons to lots of other channels. So again, psi 5, not very good here. We can derive, if you're used to using FlowJo, we can derive what they call the SSM, or spillover spreading matrix. And this is derived from the compensation matrix. So once you've derived your automated compensation matrix, you click on this SSM, and it gives a very similar pattern to this. Okay, so again, it's color coded. So I probably want to avoid channels where we have a lot of overlap, or certainly avoid co-expressed markers where we have a lot of overlap. So all these are aids to panel design. They're not going to be perfect because that is always going to depend on your particular situation. But of course, no matter how well we design our experiment, we're always going to have to get some data at the end of it, some numbers out at the end of it. So I'm not going to talk too much about controls here, apart from the FMO, or the fluorescence -1 control. So many of you will be familiar with this, and it's sort of the opposite or the inverse of our single color control. So with the single color control, I stain my cells with, let's say, PE, and I don't necessarily worry about my PE channel. I worry about the other channels. I want to see where my photons from PE end up. The FMO is the inverse of that. So my PE FMO I'll be looking in my PE channel, but my sample will be stained with everything except PE. So I can then see the contribution of the other photons in the system. So again, going back to that rain catcher cartoon I showed you earlier, if I take my unstained sample, yeah, I've got some autofluorescent background. But that's not the best control to assess true background, because the true background comes from the FMO. So in this case, these cells have been stained with the FITC marker, the psi 5, and the psi 7 marker. So as we saw earlier, some of those photons from each of those fluorochromes end up in the PE channel, and I've now only got this much space to see something that's positive, and that's what the FMO tells me. It tells me that this is my background. This is my cutoff of what's positive. So when I come in with my fully stained sample, I can be sure that anything I see above that background is really PE positive. So let me just show you one example of this in practice. So here's a slide courtesy of Mario Roederer at the Vaccine Research Centre. And this is a very bad experiment, really, and it's intentionally bad because we've used 5 different fluorochromes that are all excited by the blue laser: FITC, PE, Percy-Psi 5.5, PE-Psi 5, and PE-Psi 7. Now, if I'm looking for something that's positive in this channel, my unstained control won't have any fluorochromes on, so I'm just looking at autofluorescence. So I might be tempted to say, you know, my background is here, but that's not the right control. It's not taking into account the contribution of photons from those other 4 in the system. So this is my correct control, my FMO. It contains the FITC, the PE, the PE-Psi 5, and the PE-Psi 7 markers, but not the PE-Psi 5. This is correctly compensated, and now I see this data spread here, which is coming from, which is being revealed by our compensation. But now it tells me that actually my background and my cutoff for positivity is actually here. So remember, this sample does not have any Percy-Psi 5.5 in it. So this isn't Percy-Psi 5.5 positive. It's simply photons from the other fluorochromes ending up in that channel. So now when I run my fully stained sample, all I can really say about this is that these cells above this red line are positive for Percy-Psi 5.5. I can't say that about these. And now reviewers are much more clued up to the use of FMOs. So you're probably going to be asked for those in your multicolor experiment. So we should get into the habit of using them. It's best practice to run your FMOs probably every time you run your experiment, because they will also take into account the biological variation that you're going to get. So I said I've tried to give you a bit of an overview here of panel design. I do want to give you one practical example, a sort of thought experiment in a second, because now hopefully we've got an idea about our cells. We know where their autofluorescence is. We may know something about their size. Remember, size also influences autofluorescence. We know something about the fluorochromes we can use. And remember, not all cytometers can measure all fluorochromes that are out there, so we need to know which ones we can measure on our own cytometer. And we now know something about our antigens, both the antigen density and the expression pattern. Now, knowing all of those things helps us to hopefully combine into a great, or at least a good, panel. So in the final couple of slides, let me just give you a simple example. So we've been asked a question. We've been asked to look at the level of CD25 expression in 2 different subsets of CD3 cells. Now, you may not be an immunologist. I'm not an immunologist by training, but I know that CD3 identifies T cells. CD4 and CD8 will both identify a subset of T cells. And the question we're asking is, what's the level of CD25 on those? We're also going to add a viability marker to exclude our dead cells. So what do we need to know? We need to know our cytometer configuration. Now, luckily, that's written up here. So we know that we have at our disposal a 2-laser, 5-color flow cytometer. That's a little bit too broad, isn't it? We need to know what lasers they are. So we know I've got a 488 laser, blue laser, and a 640 red laser. Even that's not good enough. I need to know how many detection channels I have on each. So I have 3 from the blue laser, 2 from the red laser, and I also now know which optical filters are in those 3 channels. But I'm still not finished, because I want to know other things. I want to know which dyes I could potentially use in those channels, and I want to know something about the biology. I want to know what's the antigen expression level, what's the expression pattern of those 3 or 4 markers, and I want to know which cells are expressing those markers. Now you'll notice here I've got 5 colors and I've got a 5-color flow cytometer, so I've got maybe less flexibility than I might have if I were doing a 5-color experiment from a 5-laser cytometer. But the process will be the same. So let's draw that cartoon of the 2 lasers, blue laser, red laser. This is how much space I have to detect fluorescence on the blue, this how much on the red. I know which filters are in my lasers, so I can plot those there. as well. So that's the sort of spectrum covered by that 530, 30. I have 780, 60 in both of those lasers. Now I also need to know which fluorochromes I can measure in each of those filters. So I may know this immediately. Certainly if you're experienced, you would know this. Or I may go to my core facility, or I may just look at my schematic, and I find that I can, in this filter, I can measure FITC or Alexa 488 for my antigen staining. I can measure Cytox green as a viability dye. Here I can measure PE for my antibody, PI for my viability. Here I can measure PE size 7 for my antibody. My red laser, I could use APC for my antibody staining and TOPRO 3 as a viability dye. Or at the other end of the spectrum, I can use APC size 7 or equivalent for the antibody, and then either DRAC7 or one of the near-infrared live-dead fixable dyes. So I know which fluorochromes I can measure where. Now I need to think about my antigen density. Now I may know this, I may not. If I don't know it, what I tend to do is I go to Google, I type in CD3 flow cytometry, check the images. So this tells me that CD3, CD4, and CD8 have that sort of primary expression pattern that we talked about. They're either positive or they're negative. It's on or it's off. So with those three patterns, I may not need to worry about having a very bright fluorochrome. CD25 is a little bit different. It has that more secondary pattern. So although I have some bright cells, I have quite a lot of cells that are dim or just above the background. So that tells me that with CD25, I want to try and maximize my chances of seeing that by using a bright fluorochrome. Now with the viability dyes are nearly always very bright. You just have to think about the number of targets they have. If I'm using a DNA binding dye, or if I'm using a protein binding dye, there are millions of DNA binding sites or proteins in a cell. So I nearly always get a very bright signal with the DNA binding dye. So again, I don't need to necessarily worry about where that's going to go initially. But what is important, I think, even at the early stages, is to do a sort of thought experiment. So let's think about how we're going to analyze this data. In fact, I try to encourage everybody to do that before they even start their experiment. Think about how you would present this data to answer the question that you've been asked. So we're going to remove our viable cells. Sorry, we're going to remove our dead cells. We're going to gate on our viable cells, whichever way we want to do that. So I've chosen to use forward scatter here against my live dead. Within that viable cell population, remember, we're going to be defining our T cells on the basis of CD3 expression. Now, within that CD3 positive population, I want to look at expression of CD4 and CD8, and I know that they're on different subsets. Some are going to be positive for CD4, some are going to be positive for CD8. But the question I've been asked is, what's the level of CD25 on each of those two subpopulations? So this is where hopefully we come back to thinking about co-expression, because I know that on the cells that I'm interested in, they're going to be CD25 positive, CD4 positive, and CD3 positive. Okay? They're going to be negative for the live dead dye. So they're going to have 3 fluorochromes on them. This population is also going to be positive for CD25, this time for CD8 and CD3. So again, it's going to have 3 populations on it. So as well as thinking about the brightness of the fluorochrome I'm going to use in CD25, I might need to think about where I'm going to put it in the spectrum to maximize my resolution, because in both of those cases, there are going to be 2 other markers on the same cell. So first of all, I go to my fluorochromes and I rank them, or I find out the ranking. So I know that my brightest 2 here are PE and APC size 7. But if I were to use PE for my CD25, that would be here. It would be on the same laser. Remember, I have to use all 5 of these channels. There are definitely going to be 2 other fluorochromes that are on that laser. So rather than using PE for my CD25, I'm going to use APC. APC is a very bright dye, but now it's on a different laser, and there's only 1 other fluorochrome that is likely to leak into that. And what I'm going to do is I'm going to put my viability dye there, because remember, the cells that I'm interested in, those CD25s, are going to be negative for that viability dye. So the cells I'm interested in won't have any overlap from that dye. So then I can think about my other 3 markers in the blue laser. And I've put my CD4 and my CD8 in channels that are next to each other. So I know that I'm going to get probably the most overlap between FTSE and PE in this situation. But it doesn't actually matter to me because those cells are either going to be CD4 or CD8. I don't expect anything to be double positive. So I can get away with having a more spillover. And therefore I'm going to put my CD3 here at the end. Of course, this isn't the end of it. I have to then go and test this on the flow cytometer. And it may be that it doesn't work very well. It may be that I might have to swap maybe the CD8 and the CD3 out. But if I have those antibodies available in my fridge, it's worth spending that bit of time because I will get to the panel that gives me the best resolution. So we are definitely coming to the end here. So I've just slightly gone over the hour. Trying to pull this all together. So remember, we're always going to be thinking about three things. The blue things here, the instrument, the cytometer that we're going to be using. We're always going to be limited by that. In red here, we have the biology. We need to identify the antigens that are going to help us answer the question of interest. And then once we've done that, we're going to need to think about their antigen density and their staining pattern. And staining pattern also means the co-expression, because the more we think about that, the easier it's going to be to maximize our resolution. And then we can start thinking about our fluorochromes by, remember, assigning our brightest fluorochromes to our most important or our weakest markers, and thinking also about the conditions, the fixation and permeabilization, and so on. So flow cytometry is great. Multi-parameter flow cytometry has answered many questions in the last few years. Being able to add more and more colors has enabled us to devise hugely complex experiments. But with all of those experiments, we will always have to think about the tools, the fluorochromes and the cytometer. But as long as we follow the guides, remember here I tried to give you an overall impression of how we need to think about these experiments. As long as we follow those guides, we will be able to optimize it. But say be prepared to get it right. Again, if I go back to that paper, that 17 color paper, it took them maybe a year to optimize the experiment, 3 or 4 months to run the data, and then another year to analyze it. So it can take a long time. Generally, we can get a 1 color experiment up and running in a couple of days. If you're doing a 20 color experiment, we may be talking months to get that right. And of course, ask questions. You're never alone. Speak to your local experts, speak to your core facilities, and so on. So I've got a couple of questions come in, which I'll come to in a second. Just my traditional last slide here. You can find information about what's coming up, all the webinars that we're running from the CRIC, not just flow related, on our What's On page. Of course, follow on Twitter. You can follow me. The Royal Microscopical Society and Flow Cytometry UK also have some useful resources available. If you think of any questions afterwards, please feel free to drop me an email, either at my training address or derek.davis@cric.ac.uk. And at the end of this, you'll be sent a link to a questionnaire. Even if you fill one of these in before, please fill it in again, especially if you have any suggestions for any either webinars or short tutorials, which we can put up on our website. So this should take you to the YouTube link. You'll get a link to this one at the end when I put it up, which might take a couple of days. But please keep checking back, because we will put as many up there as we can. So with that, I'll just, I think we've got one question here actually. So what is the best way to titer an antibody? Can you do it with compensation beads, or have to do it with the real sample? Well, you don't have to do it with the real sample, but you can't do that with compensation beads, because what we're doing with titration, remember, is we're assessing the binding of a specific antibody to its target. So that's not what we do with compensation beads. Compensation beads are antibody capture beads. So they're actually coated with an anti-kappa antibody, so they'll bind to the light chain of your antibody. So they're not binding the specific target. So we have to do titration with a cellular target. But it doesn't have to be with your particular cells. In fact, you should do it with a cell that's expressing as many of that target as possible. Quite often that's easy to find, because the data sheet may tell you which antigen this has been derived against. So you can do it in a cell line that may be expressing that marker. I'll give you a specific example actually of that. It also depends on the conditions you're using, because we did a titration of an antibody that picks up mitotic cells. Okay, so using MPM2 to pick up mitosis. And we did the titration on normally growing cells. And of course, in normally growing cells, you only have 1% of your cells that are in mitosis. Whereas in the actual experiment, we were going to induce an increase in mitosis. So in our experimental samples, we had maybe 20 or 30 times as many targets. So the titer that we came up with our exponentially growing cells was not good enough for our cells that had more targets. Okay? So we should have done that titration on the one that had the most targets, and that's what you should always try to do. So actually, it's a very good question as well. Are there any online resources that can aid in developing a panel? Well, yes, there are. I probably should have put those in a slide here as well. So many of the manufacturers of cytometers and reagents have these online. So Becton Dickinson have one, certainly Thermo Fisher have one. And there are some independent resources as well, things like Chromocyte and Fluorofinder. So these are a good starting point because you can type in your particular cytometer, so the configuration, the number of lasers, the number of detectors in there. So that will help you, if you choose a wrong fluorochrome, it will say, sorry, you can't measure it on this cytometer. They're also linked to many of the manufacturers' reagent catalogs. So it will say, actually, this reagent is not available in this color, so you have to fit it in these ones. I think where some of these aren't quite perfect is that the staining pattern is often not taken into account with those. But sometimes we don't know that. So, but they are a good starting point. And I think in the coming years these will be refined much more. So at the moment I would use maybe a combination of those, and then actually looking at the combination they've come up with and seeing whether that really fits with your biological question and the cytometer you're going to run those on. So great, that seems to be the end of the questions there. So thank you everybody for attending. If you enjoyed this webinar, please point your colleagues and friends in the direction of these. We'll be running this and many others over the coming months. As I say, you will be able to access a version of this in the coming weeks. So once again, thank you all for coming, and we will end there. Thanks very much.